What if you can hack your DNA to make you run faster, jump higher, or become stronger? I'm Cindy, and today I will be talking about CRISPR, the gene editing tool, and how it can improve sports performance. So, what exactly is sports performance? Performance in most sports is determined by the athlete's technical, tactical, physiological, and psychological/social characteristics. This can be tested through multiple fitness tests assessing the components of fitness, flexibility, movement screening, muscular endurance, strength, power, and cardiovascular endurance. Sports performance is the manner in which sport participation is measured. Sports performance is a complex mixture of biomechanical function, emotional factors, and training techniques. Performance in an athletic context has a popular connotation of representing the pursuit of excellence, where an athlete measures his or her performance as a progression towards excellence or achievement. Sports can result in pressure on an athlete. Worrying about results, game conditions, and the opposing team can increase a sense of pressure experienced by athletes. Along with this, the athlete's ambition and drive can cause pressure to perform well, which results in poor mental clarity and wellness, which can cause worse sports performance. All athletes want to be at the top of their game when they compete, but some resort to nefarious approaches to achieve peak muscle growth, speed, and agility. So what is CRISPR? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a component of bacterial immune systems that can cut DNA and that has been repurposed as a gene editing tool. It acts as a precise pair of molecular scissors that can cut a target DNA sequence directed by a customizable guide. The system is made up of two key parts, a CRISPR-associated Cas nucleus, which binds and cuts DNA, and a guide RNA sequence, gRNA, which directs the Cas nucleus to its target. CRISPR is an important part of science because it allows scientists to rewrite the genetic code in almost any organism. It's simpler, cheaper, and more precise than previous gene editing tools. CRISPR refers to the sequences in the bacterial genome. They afford protection against invading viruses when combined with a series of CRISPR-associated proteins. Cas9 is one of those associated proteins. It's an endonuclease that cuts both strands of DNA. CRISPR spacer sequences are transcribed into short RNA sequences, capable of guiding the system to matching sequences of DNA. When the target DNA is found, Cas9, one of the enzymes produced by the CRISPR system, binds to the DNA, cuts it, shutting the target gene off. Repair after the DNA is cut may occur via two pathways, non-homologous end joining, typically leading to a random insertion or deletion of DNA, or homology-directed repair, where a homologous piece of DNA is used as a repair template. It is the latter which allows precise genome editing. The homologous section of DNA with the required sequence change may be delivered with the Cas9 nucleus and the sgRNA, theoretically allowing changes as precise as a single base pair. Cas9 is a protein which plays a vital role in immunological defense against a certain bacteria against DNA viruses and plasmids, and is heavily utilized in the genetic engineering applications. Its main function is to cut DNA and thereby alter the cell's genome. Gene or cell doping is how CRISPR can improve sports performance. Gene or cell doping is defined by the World Anti-Doping Agency as the non-therapeutic use of genes, genetic elements, and/or cells that have the capacity to enhance athletic performance. Gene doping is the hypothetical non-therapeutic use of gene therapy by athletes in order to improve performance in those sporting events, which prohibits such applications of gene modification technology, and for reasons other than treatment of disease. The impact gene doping has on sports is larger than that of normal performance-enhancing drugs because the effects can be permanent. Through this tool, athletes can enhance their speed, focus, vision, and even muscle growth. The changes are difficult to detect as the modified DNA appears as a natural part of the body. At the time of conception, when the genes from our mother and the genes from our father were combined to make us, The body then reproduces those genes that we were born with. Now, imagine if we could take an external gene that has been edited in order to enhance a muscle and that we're able to be injected into the body. The body will then reproduce that modified gene. And essentially, that's what gene doping is. The CRISPR-Cas9 system can modify DNA with greater precision than any existing technologies. An advantage of CRISPR-Cas9 is it offers other mutagenic techniques like ZFN, and it also makes it simple to amend your target region. It's very cheap, and there's already a lot of labs and publications using CRISPR-Cas9 genome editing. However, the limitations are it's not always efficient, and it can cause cardiac arrest and cancer when used to alter or enhance sports performance. The editing of the gene can also cause unwanted changes in human embryos. In my opinion, in terms of using CRISPR-Cas9 to improve sports performance, I am against it. I believe that gene doping should not be used, as if it can permanently alter any person's genetic structure to enhance their sports performance. The purpose of sports and competition would be diminished and useless. It would turn into less of a fight to strive for physical perfection through practice, but instead be a fight for who can edit as many genes as possible in their body to enhance that physical performance. This I find unnatural and straight out of a sci-fi movie. However, when it comes to CRISPR as a whole, I think it's a tool that will further revolutionize science and open new doors for us to explore what we can do with genetic engineering.